Not bad, not bad, not bad.
Really nice flavour.
Then it could exit under cell phosphate. More specific, we'll talk about fine glycolysis. You may have heard of this, but if you haven't, what does it mean? Glycolysis is an energy conversion pathway. This is the way that we take glucose, we convert it into energy, and again, just to repeat, when we're talking about energy, we're talking about the formation of ATP. This process is common across both prokaryotes and eukaryotes. So it tells you that if it's common in these two organisms, this is a very common pathway, but it's also a very important pathway. So bacteria, for example, and humans are still going to use glucose to make ATP. It is very, very important. It is going to be their preferred energy source. And it occurs in the cytoplasm. So why do we do this? We have this point here. We have a distinction of location versus organelles. Okay? So in this case, it's cytoplasmic. Later on, we'll be more specific. We'll talk about the organelle, and we'll even talk about the exact location within an organelle where a reaction takes place. Because when we come to look at the mitochondria, it is a double membrane organelle. You can actually have the inner and outer regions of the mitochondria being involved. So what happens in glucose? This is one of the first key points. Glucose is converted into two molecules of pyruvate. And remember, one of the learning aims was around pyruvate. So this is an important intermediate, one that you must know. This formation on its own leads to the generation of two molecules of ATP. This is nowhere near the maximum of what we can generate, but it's a nice starting point. Okay, so you need to appreciate that we've got some activation, we've got some energy just from this process. Now, we divide glycolysis into two stages. And so you need to know the difference between stage 1, which is up here, stage 2, which is down here. So break them up in two parts. So stage 1 is referred to as a trapping and preparation phase. This is where we take the glucose, prepare it for metabolism, but we don't yet generate ATP. So obviously, those two molecules of ATP must be in the stage 2 or the second phase of the process. For the trapping and preparation, glucose is converted to fructose 1,6-bisphosphate. So 1,6 is the location, bis means 2, and phosphate refers to the addition of a phosphate group on these two locations. So it's telling you that a fructose molecule on carbons or positions 1 and 6 has had two phosphate molecules attached. That's all it's telling you. But if you look at that number, it refers to the addition of a phosphate. Group on these two locations. So it's telling you that a fructose molecule on carbons or positions 1 and 6 has had two phosphate molecules attached. That's all it's telling you. But if you look at that name, you're the second phase of the process. For the trapping and preparation, glucose is converted to fructose 1,6-bisphosphate. So 1,6 is the location, bis means 2, and phosphate refers to the addition of a phosphate group on these two locations. So it's telling you that a fructose molecule on carbons or positions 1 and 6 has had two phosphate molecules attached. That's all it's telling you. But if you look at that name, it's really quite explanatory and it's quite definitive. So this is actually quite a useful way of explaining things. So initially, sometimes students will look at this and think, oh gosh, how am I supposed to remember this? Only those key ones do you need to remember, but you can look at this and identify what's going on. So remember, fructose is the substrate. It is phosphorylated twice on locations 1 and 6. We then look at the steps. So we've got 3 steps for this to occur. Phosphorylation step, so this is the first phosphate. It then becomes an isomer, so this is the reorganization of the location of molecules within the, or carbon atoms or groups within the molecule. And then we have a second phosphorylation step, which gives us the second biphosphate or bisphosphate group. Why is this achieved? First of all, it results in glucose that cannot exit the cell. So by doing so, it is now locked, or as we said up here, it is now trapped within the cell. This is important because once it's in the cell, it can now undergo metabolism. We can start to have energy being produced. So again, ATP. If this didn't occur, then it could exit and the cell might then be at a disadvantage and not have enough energy or enough resources later. Once it's in, it is then converted into phosphorylated 3-carbon units. So we're looking at a 3-carbon molecule, and that is an important subunit. That then moves into stage 2, and this is where the ATP generation phase occurs. What are those 3-carbon fragments, or what happens to them? Those 3-carbon fragments are oxidized. So remember, oxidation is loss, so either of an electron or of a proton, to pyruvate, and we will talk about this over and over again. It's again one of those key molecules in this process. Now, what I will do is I'll just zoom in a little bit. It's a bit of a larger diagram, a smaller diagram I should say. So, remember, we're in the cytoplasm. Stage 1, our glucose is here. We have ATP with the enzyme hexokinase, and we spoke about hexokinase previously in the cofactor lecture. So, this gives us glucose 6-phosphate. We then have the isomerization, phosphate glucose isomerase converts this to fructose 6-phosphate, and then we have a phosphofructokinase. How do we know that this is the name of the enzyme? Well, we've got a stage 2, and this is where the ATP, and it's quite definitive. So this is actually, you can the step that cannot bract. If this didn't occur, once it's in, it is then converted into phosphorylated 3-carbon units. So we're looking at a 3-carbon molecule, and that is an important subunit. That then moves into stage 2, and this is where the ATP generation phase occurs. What are those 3-carbon fragments, or what happens to them? Those 3-carbon fragments are oxidized. So remember, oxidation is loss, so either of an electron or of a proton, to pyruvate, and we will talk about this over and over again. It's again one of those key molecules in this process. Now, what I will do is I'll just zoom in a little bit. It's a bit of a larger diagram, a smaller diagram, I should say. So, remember, we're in the cytoplasm. Stage 1, our glucose is here. We have ATP with the enzyme hexokinase, and we spoke about hexokinase previously in the cofactor lecture. So, this gives us glucose 6-phosphate. We then have the isomerization, phosphate glucose isomerase converts this to fructose 6-phosphate, and then we have a phosphofructokinase. How do we know that this is the name of the enzyme? Well, we've got a phosphorylated molecule, so it's phosphate. We've got fructose, and it is a kinase. So remember, a kinase adds a phosphate to a target. So this tells us that pathway. And so we have our fructose 1,6-bisphosphate. So if I go back up, here it is, fructose 1,6-bisphosphate. This is showing you on the right-hand side. I'll just swap that out. It's showing you the same thing, but now with the chemical structures. So if you want to see what the chemical structures are, you can see where the groups are being added. You can see that this then gets converted to a different form, the isomer, and then we've got the 2 versions of the bisphosphate that's occurring. And notice we've gone from a 6 to a 5-ring sugar. You then have aldolase, which converts it to 2 different molecules, glyceraldehyde 3-phosphate or dihydroxyacetone phosphate. And we saw this in the previous lecture where we were talking about examples of these monosaccharides units. And notice, these are just isomers of each other, so they can be interconverted. And what this means is that both of these products can continue into stage 2. And again, notice some of these arrows are double-ended, so you can have some reversal up here, but not down the end. And remember, when we were talking about the control points, this is going to most likely be the control point for this pathway. So hexokinase is probably going to be the rate limiting step because it begins this entire phase. So if we can control the activity of hexokinase, we're going to control all subsequent stages. Now, if we look at stage 2, we've got glyceraldehyde 3-phosphate. So here it is. It is acted on by a dehydrogenase. So what does that do? It removes hydrogen atoms. And we can see here that we've got the phosphate NAD+, so remember one of those coenzymes, and that is converted to NADH. So 1 proton has been removed, gives you 1,3-bisphosphoglycerate. This undergoes another round of phosphorylation, so it's another kinase. A mutase, remember, converts it to, again, in that isomerized version. So we go from a 2 position to the 3 position. You've got an enolase, so you've got the release of water, so phosphoenol pyruvate, and then the pyruvate kinase converts it to our final 3-carbon atom, which is our pyruvate. You can see that here. You've got 1, 2, and 3 carbons. So all of these steps are occurring just to make this molecule. This is the one that we're interested in. And can I also highlight, here is one ATP, here is another ATP that is generated. So it's important to know ATP is generated, and it's important to know the location of these steps. Do you need to know every single one of these enzymes? No. Okay, there's too many here. I don't think it's really a useful process for you to know that, but it's important to know these key points. And the other thing that I will highlight is here that this occurs twice for every molecule. Why? Because you've got this one that can come through, and you've got this one that can come through. So this is occurring once, this is occurring twice. So hence the double output that can be generated. Okay, we'll go into more detail into that process in the subsequent lecture, but that was a lot for one starting point. So let's stop there. When we come back, we'll continue our detailed review. Okay, welcome back. So we spoke about the overview of the process. Let's go into the key stages and the key steps that you need to know. So remember stage 1. This is the trapping and the modification. So glucose is transported into the cell. This is an active process. There are glucose transporters within cells that take the glucose that is within the blood and bring it to the individual cell. We need to maintain it within the cell so that we can generate the ATP. So this is an important process. Now this occurs through phosphorylation event, and so we've got our hexokinase. And remember, magnesium, and manganese here are required. So these are our cofactors for metabolism. And this gives us one of our key intermediates, glucose 6-phosphate. So where is the phosphate? It's on the 6th position. We'll see that here. Importantly, the phosphorylation of glucose means that it can no longer exit the cell. So the transporters are blocked. Now they're blocked. What I mean by blocked is that because of the addition of that phosphate group, it interferes and it prevents it from just exiting out of the cell. Only glucose can be brought in. So it's not a physical blockage, it just modifies the shape and the structure. That phosphorylation also facilitates the metabolism into that final 3-carbon compound, of which we're really interested by pyruvate. And I want to expand on this just briefly. Why is phosphorylation so important? Because we will see this time and time again. It adds that phosphate group. What this does is that it makes the location more reactive, more likely for the reaction to take place at a specific location. So it does two things. It increases the energy because you've got a phosphate group there. So when you release that, there's an energy advantage. And also by having it located on a specific atom, you've got a specific point of reactivity. And that in turn helps for the development of the required molecule as we are moving forward. So if we look at the diagram, here is our glucose. We've got ATP being invested, so this is being used or added. So it's an energy intensive process. We've got hexokinase. Remember, hexo refers to 6, so the substrate is a 6-carbon molecule, so it's glucose. And the kinase, you can see that the vast majority of the interaction is going to be forward because this is an energy expending process. This is going to drive it forward quite significantly. This gives rise to glucose 6-phosphate, or G6P. Get used to these terminologies because, again, you'll see it in your notes quite a bit. So this is the 6th carbon, and you can see the phosphate group attached. We've now converted ATP to ADP, and we have lost a proton. So you can see this is the first step of glucose going to glucose 6-phosphate, the first step in glycolysis. And remember, this is therefore going to be a control point. This is going to be a key enzyme that we can control the whole glycolytic pathway. If we modify this, if there are other compounds that will change the activity of this, it can change the direction and also the magnitude of this activity. I've covered this before, but kinases transfer phosphates onto the substrate. In the example above, the substrate is glucose. The reaction is reversed by phosphatases, which remove the added phosphates. So if we wanted to go backwards, and here's the reverse arrow, there would have to be a phosphatase present for this to go backwards. Now, once we've done that, remember that the next stage was isomerization, changing the isomeric version. And this allows us to produce fructose 6-phosphate. So we go from glucose to fructose. So, next step is that isomerization of glucose 6-phosphate to fructose 6-phosphate. Bit of a repeat there, but that's okay. So if we look here, here is our glucose 6-phosphate. We've got the phosphate group up top, but we've now got these other molecules that are coming to play. We've got the open chain form, and you can see that it's going to want to go to the this structural form, but it can also then be interconverted into the fructose 6-phosphate form, and that in turn can also be converted into this structural form of fructose 6-phosphate, or F6P. And again, you can see that you've lost this group, and we've got a smaller grouping that is occurring. Now, this is a required step because fructose 6-phosphate can then be cleaved into 3 carbon units. And remember, this is the end result of stage 2, is these 3 carbon units. So this is going to be a really important molecule. Glucose 6-phosphate is not. So that's the reason. There's a typo there. I'll fix that. There's a reason for this taking place. Glucose can't actually achieve it, so it has to be converted into a different form. We then know that the fructose 6-phosphate is phosphorylated again to give the 1,6-bisphosphate. and it does this by phosphofructokinase. This is another one of those enzymes that you need to know because it is an allosterically controlled key regulator of glycolysis. So highlight that point. Allosteric control is at a different location, and it is a key regulator of that process. So, here is the starting point. Fructose 6-phosphate.
You see the location here, plus ATP. Notice the arrow, huge in one direction, much less in the opposite direction. One phosphate, two phosphates as part of that process. And again, ADP and the proton is released. That fructose, 1,6-bisphosphate, is then cleaved into two triose phosphates, which are the 3 carbon fragments. This is achieved by a reversible enzyme, aldolase. And these were the two that we saw at the end of stage 1. Glyceraldehyde 3-phosphate, or GAP, and dihydroxyacetone phosphate, DHAP. So, my suggestion is to read through these notes here, this written section, along with the diagram next to you, because it'll explain the steps, it'll explain the importance of those steps, and that will bring the entire picture much clearer in your mind and in your understanding. Okay, I'm going to stop there because we're going to move on in a bit more detail. And again, this is complex, so I'd rather slow down and go into shorter lectures. So let's stop here and I'll see you in the next recording. Okay, welcome back. So remember, with the previous section, we spoke about the 2,3 carbon precursors at the end of stage 1, and here we see them. Fructose 1,6-bisphosphate, this is now just acting as the substrate, and here is the aldolase. The coloration shows you the grouping, so you can see that this group goes on to make dihydroxyacetone phosphate, and again, we've got this structure here, and then we've got the remainder giving rise to glyceraldehyde 3-phosphate, or GAP. So this is those two key molecules, which is also why we say that out of the formation of one molecule, we get 2 3-carbon atoms that give rise to those 2 products of pyruvate at the end. Now, what happens with these two molecules? So, glyceraldehyde 3-phosphate is used in glycolysis, so that's fine, that's easy, can be used. But the dihydroxyacetone can't be used directly, so not used directly. So that's the first point. But what we notice is that dihydroxyacetone phosphate is an isomer of the two. So it's an isomer. So therefore, it can be converted. If it wasn't used, this would then be a waste product. And from a cellular perspective, that's a 50% wastage because you would have 2 products and only one of them could be used. This would then have to go into some other pathway. So rather than that occurring, this isomeric change allows 100% of the molecule to be used. So it is converted to glyceraldehyde 3-phosphate by a triose phosphate isomerase. Remember, an isomerase converts isomers. So it converts it from one isomeric structure to the second. So as a result, all of those products, GAP and DHAP, can now be used in glycolysis, so no wastage. So really clever system within the cell. So let's have a look at the molecule. Conversion of dihydroxyacetone to GAP allows both molecules to be used in glycolysis. So again, you can see this is a bidirectional, so it can occur in both orientations. But remember, the isomeric structure simply means that you have the same molecules, they're all there, but in a different configuration. So very useful. Now, whenever you see something in bold, like I've done here, as part of the text, apart from a heading, this is a key feature, so highlight it. The net result of stage 1 is what? You've had one molecule of glucose,
that's been converted to 2 molecules of glyceraldehyde 3-phosphate, and 2 molecules of ATP have been consumed. So it's actually generated or required energy because of those 2 phosphorylation events from the glucose to the fructose 1,6-bisphosphate. But you were thinking, well, hang on, I thought this was meant to be an energy producing process. Well, sometimes you've got to put energy into the system to then reap larger energy rewards later. And that's what's occurring in this situation. Now, we will see this time over again, this particular process. So that's stage 1. Let's now move on to stage 2. So remember, we've got 2 molecules of glyceraldehyde 2-phosphate that have been converted or been produced. This now needs to be converted into the next substrate for it to participate. It's converted to 1,3-bisphosphoglycerate. So again, 1,3 is the location of 2 phosphates on a glycerol backbone. The glycerol is the 1, 2, 3 carbon atoms that we have here on the left-hand side. Again, this is a redox reaction. Remember the cofactors that we have NAD+. This is going to take a proton, it's going to take this proton, and we need energy, which is the phosphate. So it goes forward, the phosphate is replaced at that proton, the proton goes to NAD+ to make NADH, and we've got the release of another proton. So 1,3-bisphosphoglycerate as that molecule. And I've even reminded you, note the involvement of NAD+ and its reduction to NADH. Why is this molecule so important? This molecule, so 1,3-bisphosphoglycerate, is energy rich. And this is the molecule that is used to generate the ATP. So again, another key molecule in this process. So notice, I'm trying to highlight to you those key steps, those key enzymes, and in particular those key molecules, and why they are key. Try to eliminate all of the other things that are less critical, but focus on those key issues here. So phosphoglycerate kinase transfers the phosphate from 1,3-bisphosphoglycerate to ADP. And remember, ADP has got 2 phosphates. To make ATP, we need to add more because this has got 3. And so that's what's occurring here, ADP to generate ATP. Now, 2 in total are generated since we've got 2 molecules of glyceraldehyde 3-phosphate are generated because we started with those 2 molecules at the beginning. And so you can see that here. Bisphosphoglycerate, here is the phosphate, here is our ADP. The kinase takes this phosphate, transfers it to ADP to now generate our ATP. So this is now energy. And here is our 3-phosphoglycerate because we've still got another phosphate at this location. Now remember, stage 1, 2 ATP were consumed. We have now generated 1, so really we've only used up 1 ATP because we've been able to generate another one here. What's the next step? We convert the 3-phosphoglycerate, which is the previous molecule, to 2-phosphoglycerate. And this is through an enzyme called a mutase. Now, as it says down here, what is a mutase? The mutase simply changes the location of the phosphate on the 2 molecules because the 2 molecules are isomers. So you can think of it like an isomeric chain. The enolase that we'll see here introduces a double bond, and by doing so, we have the release of the phosphate group occurring more readily here. So 3-phosphoglycerate, here's the mutase. Notice that we go from this location, and all it does is shift to a different location. Very simple change. Why? Because this makes it more reactive so that the subsequent change is going to give us a product. This is a hydrolysis reaction, so here is the water that is coming off. The phosphate is attached, and we've now got this carbon-carbon double bond. And you can see here that we're going to start to have to lose some components. So ADP plus a proton gives rise to another ATP through pyruvate kinase, and we generate our our pyruvate. And we've got our 1, 2, 3 carbon molecule that we are going to need to make energy later, additional energy. But again, you can see another ATP molecule that has been generated. So at the moment, we're even balanced because we've used 2 in stage 1, we've generated 2 ATP in stage 2. So as we said, an enolase converts 2-phosphoglycerol to phosphoenol pyruvate, which is here, and that is then converted to pyruvate. The loss of the phosphate from phosphoenol pyruvate is used to generate another molecule of ATP, which we saw here. But don't forget, there are 2 glyceraldehyde phosphate fragments that enter stage 2, so actually the reaction is double. So I've only been focusing on 1, but we know that we do this twice, so we're going to actually be ahead with the ATP that is generated. So if we look here, what is the net reaction? 1 glucose molecule, we use 2 phosphates, 2 ADP, and 2 NAD+. This gives us 2 pyruvate molecules, 2 molecules of ATP, we've got 2 NADHs, 2 protons, and 2 water molecules. So hence the generation of the ATP. So an important process. Now again, don't get overwhelmed by the steps. Think them through logically. You can see the process. All that we're trying to do is to either change the structure so that it becomes more reactive, and what we are interested in is just to generate this molecule here, pyruvate, because that is going to be the molecule that is important, and we'll see this in the subsequent recording. I'll stop here because we're now going to focus on what happens to the pyruvate in the next recording. Okay, welcome back. So we finished off by talking about the net reaction, looking at pyruvate. So as the title of the next section talks about, what happens to the pyruvate? What is the fate of this pyruvate? Why are we so interested? Now, for glycolysis to proceed, NAD plus must be regenerated. And we saw that we're starting off here with NAD plus. So this has to be regenerated because it's actually present in limited amounts. And remember that when we were talking about cofactors, that these are generally regenerated, and therefore the cell doesn't require high levels. Because if you can recycle, there's no point making lots and lots of these molecules if at the end of the reaction you're just recycling it back. So there's going to be a limited amount. Now, what happens to pyruvate, or the fate of pyruvate, actually varies depending on the organism that we're actually talking about. And we'll talk about yeast, and we'll also talk about humans in this process. So it's important for you to understand the difference. Pyruvate can be converted to ethanol, lactate, and these are both fermentation events, or to carbon dioxide and to water. What dictates the formation of these molecules, or the choice of these two molecules, so the production of ethanol or lactate, is the presence or absence of oxygen. So in the absence of oxygen, we generate ethanol or lactate. So ethanol is something that yeast will do, for example, so as part of fermentation. Lactate is something that we can do as humans when oxygen is limited. So you'll hear this all the time when someone is exercising and there is a buildup of lactic acid because there's not enough oxygen getting to the muscles. In the normal situation where you have plenty, so in the resting state where you've got plenty of oxygen, so in the presence of oxygen, it's going to be converted to CO2 and water as the end products. And remember, this is what we exhale, and water just becomes part of our matrix because obviously we're very much water component organisms. So the 3 pathways, and you need to know these 3 pathways, and they're all dependent on the oxygen. So pyruvate goes to acetaldehyde and ethanol, and notice the regeneration of NAD plus here. It can go to lactate, again, regeneration of NAD plus here, or it can go to acetyl-CoA, and this then allows it to undergo further oxidation, and this is where we get the bulk of ATP generation. So know those differences, know those 3 pathways. Now, in yeast of a generation. So know those differences, know those three pathways. Now, in yeast, ethanol is formed from pyruvate. So here is pyruvate, see the structure, pyruvate decarboxylase. Whenever you see D, you have loss. It is taking something away. It's telling you carboxylase, so that refers to carboxyl is going to be a CO2 unit. And you can see that here, that we have this carboxyl group, COO, is going to be removed, and it is lost as CO2. This remaining small structure is acetaldehyde, because we've got this aldehyde group, and we've then got NADH, plus a proton, we've got another alcohol dehydrogenase. Now, in this case, the dehydrogenase is adding to the carbon atom, well, actually not there, wrong location, sorry, to the carbon atom here, and we've got the proton being added. The valency of the carbon is now back to 4, so we've lost that double bond, and this is ethanol. So, as it says here, note the regeneration of NAD+. This is an important concept. What about humans? As I mentioned, pyruvate can be dehydrogenated to lactate in humans. So we've got pyruvate, we've got NADH, and the proton, we've got lactate dehydrogenase. And so we lose that oxygen. No, sorry, again, mistake. We add that proton here, and so we end up with that triple balance. So we've got 1, 2, 3 here, the proton is on the other side, we've got the hydroxyl group here, and then the hydrogen atom there. So this gives us lactate. And again, we're regenerating NAD+, so this is important. Now, as I mentioned before, we were talking about muscle. So remember that you've got fast and slow twitch muscle. This is really just referring to their ability to contract. So fast twitch muscle can work in the absence of oxygen, but just for very short periods of time. So when you look at Olympic runners, so Usain Bolt as an example, those runners have a lot of fast twitch muscle. So they can work and run really quickly, but for very short periods of time, so under 10 seconds. So they're running for 10 seconds because they can operate in the absence of oxygen for that short period of time. But because there is no oxygen, we end up with the formation of lactate in the muscle. And that gives us that characteristic burning sensation that we have all felt. It also regenerates the NAD+, and this occurs through the reduction of pyruvate to ethanol in yeast or to lactate in humans. And this is what allows for glycolysis to occur under anaerobic conditions. Otherwise, it would stop. And so, unless oxygen was plenty, this wouldn't be able to continue. And obviously, with exercise or exertion for a human, this would become problematic. However, for full energy release, pyruvate has to enter the mitochondria. So, an important idea here. It goes from the cytoplasm, has to go to the mitochondria. This has to be converted to acetyl-CoA, and this is one of those key intermediates of metabolism. This then enters what is called the citric acid cycle, and this is where we generate the huge amounts of ATP. So we've got the citric acid cycle, we've also got the tricarboxylic acid cycle, or we can call it the Krebs cycle after the scientist that worked it out. These are all the same. I think in my lecture notes I just call it the citric acid cycle, but if I do slip up and talk about one of the others, you know what we're talking about. We're talking about the same idea. And the entry point into the citric acid cycle is acetyl coenzyme A. Again, another one of those cofactors, but it's taking the acetyl unit. And again, the previous lecture, or the previous two lectures when we spoke about cofactors, I focused on coenzyme A because of the important role of acetyl units. So now you can start to see how these are all coming together as part of this energy process. Okay, I'm going to move on to other sugars, but again, I want to stop here, take that information on board, make sure you understand it, because we've got those three pathways that are important, and we'll come back to the next part in the next lecture. Welcome back. So we've been focusing on glucose, but remember the learning idea was how does the cell metabolize sugars other than glucose, because we can take other sugars within the diet. So two common examples are fructose and galactose. These are commonly found in our diet. And what we find is that there's actually no direct pathway for these sugars. So there has to be a conversion step. They have to be converted to intermediates that can ultimately enter the pathway. So it is a more involved process. And why? Because remember, the cell is adapted to using glucose. So other sugars, yes, can feed in, but its preference is always going to be for glucose. So if we see here, we start off typically with glucose to give us glucose 6-phosphate. Galactose can be converted into glucose 6-phosphate. And we saw previously when we were talking about carbohydrates that galactose was 2 glucose units joined together. So that's one way. We've got fructose from adipose tissue, so as fat stores can feed into the fructose 6-phosphate step. So remember, we had the interconversion because this was a requirement. So again, that's another pathway of entry. We've got the fructose 1,6-bisphosphate. So again, we've now just got another way of increasing this molecule. Oops. And then we've got our two intermediates, dihydroxyacetone and glyceraldehyde phosphate, and these go through to make pyruvate. You can also have involvement of fructose through the liver in both pathways to feed into these molecules here. So it can be higher up, it can also be lower down, so we've got these entry points. So nothing is going to be lost, which is useful from our perspective. So as we mentioned before, Fructose can also be phosphorylated to give us fructose 1-phosphate. We've already seen that that's a key intermediate in glycolysis through the action of fructokinase. GAP and DHAP are produced, and DHAP has to be converted to glyceraldehyde phosphate for glycolysis, so nothing is new there. Galactose is converted to glucose 1-phosphate in 4 separate steps. Once that's occurred, it can then continue within the pathway as for glucose. Now, I haven't shown you those steps because really the focus is not on galactose or other sugars, it's really about glucose. So the key point of this part of the lecture is how do other things come in. So the key point really is this diagram, because it shows you how you can have it into conversion. The steps that are occurring here, don't worry about it. It's not critical. It's just this is being converted into this is converted into here. Here it's a direct conversion, it's just a phosphorylation. Here we're going from one molecule to another, hence the addition of steps. But really the focus is just going to be on the glucose and it's moving into these other pathways or different derivatives. So hopefully that helps to reduce some of the load. Okay, so we've spoken about the generation of the pathway. And remember that I said to you it's important to understand the pathway, but it's also important to understand the control of pathways. So now what are the control of pathway points of glycolysis? So first of all, there are 2 roles of the glycolytic pathway. One is that we have glucose being degraded to generate some ATP. Remember this was in stage 2 as we've covered before. But the other is providing building blocks for the formation of fatty acids and amino acids. So this is biosynthetic reactions. So it doesn't all have to go to energy. It can also go to the formation of these other molecules through a biosynthetic pathway. So the rate of conversion of glucose to pyruvate is regulated to meet both of these needs. If you need a lot of energy, the cell will change its metabolism to focus on generating products that give ATP and also the acetyl units that go into the citric acid cycle, so the pyruvate, and then its conversion to acetyl units. If, on the other hand, the cell is growing, there's a requirement for fatty acids or amino acids, it can shuttle across to the other pathway. So what we find is that enzymes that catalyze irreversible reactions are the areas where we've got potential sites of control. Because if we can regulate at these points, we can then dictate whether we go into part 1 or part 2. So making ATP or building other molecules. So what are these control points? And these are the ones that you must know. It's going to be phosphofructokinase, hexokinase, which we saw was right at the beginning, and pyruvate kinase, which was right at the end. All of these are irreversible reactions. The activity of these enzymes are controlled through allosteric effectors. And remember, this was at an external site, and we can either increase the activity or we can decrease the activity. This is a really important idea. You should be familiar with allosteric control already, but remember, you can think of it a bit like a dimmer switch. We have the ability to turn the enzyme activity up, and when we've got more activity in one pathway and we don't need it, we can turn it down. And shuttle those intermediates into other pathways. And as a result, we'll end up with a different outcome. And we've got multiple control points. Here we've got the phosphofructokinase, hexokinase, and pyruvate kinase. Notice these are all kinases. Okay, so it's all going to be phosphorylation events. And remember earlier we spoke about muscle versus liver. So now we're going to start off with muscle as the control point. So, phosphofructokinase tells you the kinase that acts on phosphate that is attached to fructose, so PFK. This is the most important control point in the mammalian glycolytic pathway. So immediately that tells you this is an important point for you to understand. High levels of ATP, allosteric, inhibit PFK. And this makes sense because the activity of this is going to generate ATP. So if we have high levels of ATP present, we don't want this enzyme to be active because there's already an excess of ATP. So here we've got a negative feedback loop. Goes back, turns off the enzyme. It does so by lowering the affinity of PFK for its substrate. And remember, the affinity is just a measure of how strongly it binds to the substrate, how little needs to be present for the bonding to occur. So high ATP means that energy requirements are going to be low, as I mentioned before. In contrast, if we have a low pH, so an acidic pH, and this occurs when we've got a lot of lactate, so when the muscle is contracting anaerobically, this is going to signal to the cell, there's lactate, oxygen is missing, we need energy, we want to turn off this particular pathway too, because we're going down the wrong pathway, we want the oxygen to be present. So it's another way of inhibiting it. And what this does is that it prevents muscle damage because we don't want to keep producing the lactate because oxygen is obviously essential for the cells to survive. And also that acidity is not going to be good to the cell. So this diagram here is showing you how PFK can be allosterically inhibited by higher levels of ATP. So we've got velocity on the vertical axis and we've got fructose 6-phosphate on the horizontal axis. So in low ATP, we see a normal Michaelis-Menten kinetics. As substrate goes up, we get increased velocity, and then here, we've reached the plateau region. But notice that at very low substrate levels, if we've got high ATP, it's very much delayed until we get to really, really high levels and then it catches up. But notice there's this massive gap of reduced activity, and we still are not reaching maximum activity, but we have to be at really, really high concentrations of the substrate. So this is showing you that inhibition that is taking place. If we then move on to the next step, the hexokinase, remember this catalyzes the very first step in glycolysis, and I mentioned previously that the first step is usually the rate limiting step, because if you can control that first step, you control the entire pathway. It too is inhibited by its product. So remember, we generate glucose is converted to glucose 6-phosphate of PFK for its substrate. And remember, the affinity is just a measure of how strongly it binds to the substrate inhibition, or that negative loop that is occurring. And so this prevents the production or overproduction of excessive substrate. And lastly, pyruvate kinase, this controls the efflux from the pathway. Remember, this is going to generate the pyruvate. So again, ATP allosterically inhibits pyruvate kinase, and it's for exactly the same reason as phosphate fructokinase. If there is already a lot of ATP, there's no point taking that substrate to generate more ATP. So we want to turn off the pathway. So let's look at these diagrams. So diagram showing the control points of the glycolytic pathway. So first of all, highlight here at rest. So this is now us. We're sitting here, we're reading, we're listening, we're heavily concentrating, we're at rest. So glycolysis is inhibited because the energy requirement is quite low. What we see here is the entire pathway. So there's nothing new. And what we have here is the muscle fibers are relaxed. Because of that, there is no build-up of lactate. And remember, there's no acidity going up, so there's no marker. So as glucose is converted by hexokinase to glucose 6-phosphate, this level comes back, negatively inhibits and stops this from occurring. Whatever glucose 6-phosphate is there, if there's excess amounts, it gets shuttled and it gets converted into glycogen, which is a storage form of glucose. So we don't go through this entire pathway. So all of these subsequent steps are going to be prevented because this is going to be limited. Most of it is going to glycogen. If we keep going and we say, well, okay, we've got a lot of ATP or low levels of, or the ratio between the two, you see that ATP comes back and blocks phosphofructokinase, again, stopping it going forward. And right at the end, pyruvate kinase is also blocked by ATP to stop the formation of pyruvate. It's the same process that's occurring. So we've just got multiple points within the same pathway. Because remember, we've got other ways to feed in. We can feed in fructose here as well. So we've got to have a bypass. If we bypass glucose but we just have fructose, then this is going to be overactive anyway. So that's not going to be useful. So it's important to have multiple points of control. The other thing to highlight is that we've got small, thin arrows. Why? Because if we look over on this side, we can see that the arrows are wider, showing that there is a movement. So here we're showing that there's really not a lot of movement. Over here with the large arrows, this is indicative of a lot of movement of products through this pathway. So during exercise, there is energy requirements. There's going to be build-up of lactate if there's oxygen is limited because we're unfit or that we're exercising at a really high level. So therefore, glycolysis is going to be stimulated. So what we see now is the reverse. Glucose is acted on by hexokinase to give us glucose 6-phosphate and keeps going. Notice now that glycogen can feed into the pathway. So it feeds in as opposed to before. And again, this should make sense. This is here as a short-term storage. So as it's required, it's another way to feed into the pathway. We notice now that ATP, or the ratio, is now reversed. So high levels of AMP. So ATP inhibited, AMP stimulates phosphofructokinase. Fructose 1,6-bisphosphate is generated. It now stimulates pyruvate kinase. Because if this is higher, it needs to make its way through the pathway. And as a result, the kinase gets activated. And so we generate more pyruvate. And we either use lactate for a sprint, or for the long term, we have the normal metabolism which is occurring. So we end up with CO2 and water being generated. So a very low, more gentle process where there's lots of oxygen. So oxygen plenty, oxygen is low here. And you can see here the muscle fiber is showing contracted, so it's activated. So it's contracting, and so there's energy requirements that are occurring within the cell, or within the host. Okay, that's the muscle. Let me stop there because when we come back, we'll focus now on the liver because it is going to be slightly different. So see you in the next recording. Okay, welcome back. So we finished with muscle. So now let's move over to glycolytic control in the liver. So first of all, I want to talk about why we're focusing on the liver. First of all, the liver has great diversity in its biochemical reactions. So it's involved in metabolism, controlling lots of different processes, but in particular, it maintains the blood glucose concentrations within the organism. It also has other metabolic and cleansing functions, but we're going to talk about it in the context of glucose. It is also the organ that is responsible for storing glucose as glycogen and then releasing it later as stores are low. And so all I'll say here is see diagram above. So we saw that here where we had the release. So this was going to be the liver coming into play, allowing for glycogen to be released. And again, we've also got the liver coming into play here for the storage. So you can see that these are not going to be exclusive. They have to work in tandem. So it's important that you understand how they control and regulate each other. So phosphofructokinase. ATP and changes in pH are going to be less important for controlling phosphofructokinase in the liver. And this makes sense because remember, those changes in ATP and pH are within the muscle where you've got this active metabolism. So it's going to be dependent on other byproducts for its control. So what we see is that PFK is inhibited by citrate, and citrate is an early intermediate of the TCA cycle. And here's an example where I have used a different wording, citric acid cycle or the Krebs cycle. This is one of those early intermediates, which remember, is where we start to generate ATP, or going down that pathway. So citrate is one of those molecules. So therefore, if we've got high levels of citrate, this means that there are high levels of biosynthetic precursors. So extra molecules are not required because they're already feeding in. So as a result, the liver is going to respond to these changes in blood glucose, and it can do this through the fructose 2,6-bisphosphate. Remember, that was that control point. We go from glucose to fructose, so it's a key molecule, and we said that previously as well. So in the liver, fructose 6-phosphate levels rise when glucose levels rise, and this leads to fructose 2,6-bisphosphate being produced. That in turn activates PFK, which in turn stimulates glycolysis. It's got to go through the pathway. So if we see this diagram here, again, look at the arrows. These are big and thick arrows showing the flow of molecules. Glucose to glucose, fructose 6-phosphate. Some of it gets cycled across to fructose 2,6-bisphosphate, which activates phosphofructokinase and gives us the 1,6-bisphosphate. So diagram showing how fructose 2,6-bisphosphate controls PFK in response to blood glucose levels for the liver. Okay, so let's have a look at this diagram. Let me just zoom out so we can see it a bit more clearly. So relative rate of fructose 1,6-bisphosphate synthesis, and here is the substrate, fructose 6-phosphate. So what we've got here is fructose 2,6-bisphosphate. You can see that in the presence of fructose 2,6-bisphosphate, which is here, fructose 2,6-bisphosphate, we say it activates. You can see that just 1 micromolar gives you a massive spike. At very low levels of substrate, we get massive activity, and very quickly are we at a plateau level. We don't even need a huge amount. Notice even 10-fold less, 0.1 micromolar, gives us still this massive increase. There is a shift, but it's not significant. It was not enormous. But in the absence, notice that there is this market delay. So we need much higher levels before we start to have this increase. So, as we can see here, diagram showing how fructokinase is activated by low levels of fructose 2,6-bisphosphate. And on the other side, we've got ATP stimulating activity, but then it changes to an inhibitor. So here we've got a bit more of a complex idea. At low ATP levels, we want to stimulate the production, but remember, ultimately we're going to reach a threshold. And as we do, it then starts to come down. But notice that as we start to have fructose 2,6-bisphosphate, the magnitude of activity gets higher, so on the vertical axis, and also the shift to the right also increases, so we get greater production. ATP still has an effect, but we need much higher concentrations. You can see here. So here we're about half a millimolar, whereas now this is 1 millimolar, and here we're about 1.5. But notice the tail. There's still a lot more activity, and certainly the area under this curve is much higher, showing that stimulatory effect. If we now look at hexokinase, this is also a regulatory enzyme in the liver. But the enzyme responsible for glucose phosphorylation is glucokinase, not hexokinase. There is a difference. So this is one of those differences to remember. Glucokinase for the liver. It is an isoenzyme of hexokinase, so it is a variant form. So as I said here, it is a different gene and amino acid sequence, yet it has the same function. Glucokinase has a different Km. So remember, Km is the substrate concentration that gives 50% activity. And as a result, it's going to be less well, its reactivity will be different to hexokinase, and it only works when glucose levels are high, so after a meal. So the substrate level has to be much higher for glucokinase than we would expect for hexokinase. It is also not inhibited by glucose 6-phosphate, as hexokinase is, as we saw in the previous diagram. So there's no feedback loop in this case. So again, this is another difference. And pyruvate kinase we have here, an isoenzyme variant exists for the liver. There is the L form and the muscle, which is the M form. And having the letters makes it easy to remember. So there are differences again. Same control for both, except the L form is inhibited by alanine, which is an amino acid. Why are we talking about alanine now? Alanine is made from pyruvate. So therefore, this indicates that building blocks are available. So remember, we spoke about fatty acids and amino acid synthesis before. This is an example where we've got biosynthetic pathways. When these are being built, it tells you that there is plenty available. So this L form, so the liver form, but not the muscle form, is controlled by reversible phosphorylation. So adenine phosphate does something, either stimulates or inhibits. Typically it's going to stimulate, and therefore the removal of it is going to turn it off. It's going to have the opposite. So as we see here, when blood glucose is low, pyruvate kinase is phosphorylated, inhibiting its activity. Now think about that. If glucose is low within the blood, we don't want the pyruvate kinase being activated, because that will funnel more of it through, depleting the glucose that would be present. On the other hand, when glucose levels are high in the blood, it becomes dephosphorylated, returning its full activity. So this is one of those examples where actually adding the phosphate group doesn't turn it on, it turns it off. Okay? By and large, we will see predominantly that when we phosphorylate, we normally have stimulation, but this is a good example of where it is the opposite effect. So let's have a look here. Diagram showing how liver PK activity can be controlled by the addition, which is inhibition, and then the removal, which is activation of a phosphate group. So, high blood glucose, we've got the phosphorylated kinase. If we remove the phosphate, we dephosphorylate, it becomes more active. If we add it, it turns off. So this is that cycle that we're talking about. Here, we've got the precursors, so phosphoryl pyruvate, fructose 1,6-bisphosphate as a positive regulator, ATP and alanine as negative regulators, looking at pyruvate. So remember, if pyruvate was high, we go down this pathway, we turn it off. If pyruvate is low, or these are high, we go down this other pathway. So there's this beautiful control mechanism that exists that allows for this control to take place. Okay, a lot to cover in that lecture, but hopefully I've broken it down into very small segments. Make sure you go through your chapter summary.
Okay, so apologies, I'll just repeat that. For everyone watching the recording afterwards. I forgot to press record, so we've gone through that first question, which is outlining the roles played by carbohydrates. So, major class of, one of the four classes of biomolecules, important energy source, fuels, metabolic intermediates, and these are broader thinking points, as we'll see later, that carbohydrates play a role in RNA and DNA. So there you've got a sugar, but involved in nucleic acid. Structural functional roles, so cellulose, for example, in the cell wall, and also being linked to other molecules like proteins and lipids, which are important for their function, but also for their localization in the cell. So this is now more of a chemistry question. Why is the beta chair form of glucose more stable? Think about the structure. This is about the representation of glucose and how it's drawn, and think about the groups and how they might interfere. Okay, oh, excellent. So mainly reduced steric hindrance, favorable spatial arrangement of substituent groups, and less steric hindrance. Perfect. So what you need to, think about less steric hindrance between the groups, therefore it's more stable. So you see here the chair form, you've got these larger groups. You can just see here that when this boat form is brought together, those groups are brought closer together. There's going to be charge interactions, there's going to be electron cloud interactions that are going to make that less stable. And so therefore the chair form is going to be the more stable form. You've got things spaced apart, and that provides greater stability for that molecule. This is not just the case for glucose. This is going to be true, obviously, for other molecules as well. And we saw that with proteins. As part of the three-dimensional shape, the folding, the structure, it was all about the stability of those different groups. And in turn, that's what conformed and led to the lowest energy state. So this is just another example of that. Okay, excellent. How are monosaccharides modified within the cell, and why is this process important? So over to you. Let's start off with the first one. So glucose as the prototypical carbohydrate, just as the example. So remember, you've got the different groups. So you've got reactions between alcohols, amines, phosphate, increased diversity, increased biochemical versatility. We know that there are alcohol groups on glucose. We can see that here. We've got a hydroxyl group. We've got amines on proteins. We've got phosphates that are on different molecules as well, and that leads to that interaction. And so this helps with that chemical reaction. And again, what we're asking you to think about here is that yes, it's a sugar, yes, it's glucose, but it has functional groups. It has reactive groups, just like any other group. If we took away the word glucose, we said, here's a molecule with these reactive groups, and we looked at it purely from a chemistry perspective, you would say, well, here's a reactive group, or here's a hydroxyl group, this can form a bond, here's an amine group, this is going to be able to form a bond, etc. And you would think about it in the same way. So I think that's perhaps where some of that variation comes into play. And this really links into what we said before, that these variants then have other functions within the cell. And so we set that up here, where they have different activities, particularly metabolic intermediates, but here we're talking about signaling and other metabolic fates of these sugars. So these molecules can be generated within the cell. They're not part of energy, but they're actually used within the cell to generate signaling or to transmit signals within the cell. Okay, and the last one, which is a fairly simple one, storage form of glucose in animals versus plants. What do you think? Amy, animals glycogen and plants are starch. Perfect. Animals glycogen, plants are starch. So same thing, remember, polysaccharide of glucose linked together. That is these two here, glucose 6-phosphate, dihydroxyacetone phosphate, we'll see in the next lecture, where we're looking at these as intermediates. The bonds, 1,4, 1,6, so we saw that with the glycogen, and then the summary. Okay, so over to you. Are there any other questions or comments for this lecture, or is this chemistry? This is biochemistry, but you'll notice that a lot of the emphasis that I put on the chemistry is not really in terms of the structures or remembering any of the reactions. It's more about the enzymes. It's more about the key enzymes as key control points, and really the processes, what is going on. And again, if you think back to the learning objectives, you think back to these tutorial questions, you should see that they're aligned. Okay, so when the chemistry is included, it is there really as a demonstrating aspect to try and explain a process, to explain what is going on. Unless I tell you really clearly in the lecture, in the tutorial, this is something that you must remember. Most of it is there as additional content to explain it, put it in context. Okay, so please don't go into this and think, oh, I've got to remember all of these structures and be able to draw these groups, unless it's something critical. And really the only one I can think of is probably ATP, but even that, it's not super critical in terms of drawing a structure. It's more about what are the groups, where are the functionality, where is the point of reactivity. Okay, and even with glycolysis, what's the point of glycolysis? It's really quite simple, just taking glucose and converting it into some energy. How do we use glucose to make energy? And the energy is always ATP. This is the first step in that process, right? You'll see that when we go through this, you'll see that it can be energy intensive, that you have to put energy into it to get energy out. And you'll also see that the amount of energy that you get is, in these initial stages, not fantastic, it's not huge, right? But this is not the main point. The main point is obviously when you come through to the other cycles, when you get into the citric acid cycle, that is where you get the bulk of the energy. And what you'll also see is that there are lots of key intermediates that we will talk about time and time again. So pyruvate, acetyl-CoA, etc. Those molecules that we talk about feed into the system. The pyruvate feeds in, and the acetyl-CoA is one of those molecules that goes into the citric acid cycle. But importantly, those molecules can also be used for synthesis. So even though we're talking about this from the perspective of energy generation, don't lose sight of the fact that we're not always making energy. You do need to grow, you do need to build things. And so those molecules can then be used to make energy, or to generate energy, but can also be used to generate other molecules within the body that are required. So it's important you understand the flux and the movement. Now, what you can see up on the screen are obviously the questions, but what I want to do is I'll go through the questions, and then I want to go back through the lecture notes, because I've highlighted different sections in yellow to highlight to you what I think are the key points. Okay, I think that's probably doesn't really matter which order we do it in, but I think that's the order that I'd like to start off with. So, over to you. Why is the metabolism of glucose significant in the context of energy production? And here I've said think about different organs within the body. We know it's energy, but why is it glucose? Both of you. So, only fuel source for brain under non-starvation and for red blood cells. So, really, really important. It's the only fuel. So if it's the only fuel, the body is locked in to that source as an energy source. Other tissues can use other things, but if this is the key one, then this is going to be critical. Okay, so we know now why glucose is important. So if we've got glucose, then we need to start to think, well, how do we get energy out of the glucose? And this is what the glycolysis process is talking about, is those stages, that initial starting point. So we break it down into stage 1 and stage 2. And this is where you need to start to think about, okay, if I've got this molecule, stage 1 is all about how do I get it into a form that can be used by the cell, that can enter a metabolic pathway. The second stage is really going through that process, okay, of taking those molecules, generating some energy, generating some outputs, some of those cofactors that are being generated, and that's really important as well. We'll start to see those cofactors coming into play. And then those molecules that are generated are then going to feed into the next part of the pathway, where the bulk of the ATP is going to be generated. So, key events and enzymes involved in stage 1 glycolysis. So notice the 2 points here. Key events, which is really what I've summarized to you just there, and what are the key enzymes? Key enzymes are always going to be either the first enzyme, because it's the first one, it's the rate limiting step, or those that are important for allosteric control. Those where you've got a product that comes along and controls the rate. Typically it's going to be the first one, but it can also be other points where you've got a reaction that might be reversible or irreversible. And so if you've got a reaction that is irreversible, that again becomes the point of control. If you've got something that can go both ways, well, it can flux. Reagents can move in both directions. If on the other hand you've got a point where it stops, then it limits what can go back. So if things move to that point, then if they wanted to go back, well actually you can't, there has to be an alternate mechanism for that to come into play. So if I look at the first point, trapping and preparation phase. This is all about preparing the glucose for entry into the system, into the process. And that's why it says there's no ATP generation. And in fact, you actually have to expend energy for this process. Now, a nice way to think of this, at least in my mind, this is similar to what we do if we use, say, aluminium as an example. You want to create an aluminium can. What do you have to do? Well, you can't just go get it out of the ground. You've got to mine the ore. You've got to spend a lot of time and energy to put into finding it, converting it into a form that can be used, putting energy in to then generating a product that is of benefit to you afterwards. It's the same sort of idea with the glucose. We've got to get the glucose, so we ingest it. We've got to then convert it into a form that goes into the cell and stays within the cell because we don't want it leaving. And then we've got to modify it so that it can actually convert into a product that fits into the metabolic pathway that then feeds in so that we can generate energy. Okay, so what are these first stages? What do you think? And remember, if you're happy to, more than happy for you to unmute yourselves. Gets converted to ADP, and so that level is also being monitored by the cell. The enzyme hexokinase. Remember, kinase is an enzyme that transfers phosphate on to a substrate, so you know what it's doing. And hexo means 6. How many carbons are in glucose? 6. It's really quite informative. It's really telling you what it is doing. So hexokinase is the one that's doing that process. It becomes glucose 6-phosphate. Again, that makes sense. And then it has to be rearranged to get into a different form using an isomerase. And what you'll see is that there are different forms of these products that are generated. So thank you. So glucose converted to fructose 1,6-bisphosphate. So this is a later stage. So phosphorylation, isomerization, and that other phosphorylation step. So it's that processing pathway. So as I mentioned, results in glucose that cannot exit the cell. There are channels within the cell and transporters that allow glucose to enter. And so once it comes in, you don't want it just going straight back out. You want it to be within the cell and to be trapped. This phosphorylation traps it within the cell. It can't exit because of that charge difference. And so it's now staying within the cell. It can then be converted into 3 carbon units. If we think about the citric acid cycle, right, which is much later on, citric acid, the alternate name is tricarboxylic acid, or TCA cycle. Tricarboxylic, tri for 3 carbon. So 3 carbon units are what going into the process. So what do we see? We see 1 molecule of glucose is converted into 2 molecules of glyceraldehyde 3-phosphate. And you know, I'll get to the diagram later, there's 2 molecules of this that are generated, but there's also another molecule that is made as a byproduct. It is an isomer. It is then converted into generating 2 molecules of glyceraldehyde 3-phosphate. And we've had to consume 2 molecules of ATP in that phosphorylation process. We've had to phosphorylate it multiple times, and we've had to expend energy. We've had to put energy into the system. Okay, perfect. So key events then and enzymes in stage 2 of glycolysis. What do you think? GAP is now converted to 1,3-phosphoglycerate by redox reaction. Phosphoglycerate kinase transfers phosphate from 1,3-phosphoglycerate to ADP to make ADP. Reduction to NADH takes place as well. Okay, great. So redox reaction. Remember, metabolism is redox. You can see that we've got different reactions that are taking place. We've got more phosphorylation, and we've also got the role of NAD+ going to NADH, which is part of that redox reaction. Remember, redox is reduction oxidation. It can be either electrons or it can be protons. So NAD+ takes the proton, becomes NADH. And in taking that NADH, you can also donate that proton. It's able to interconvert. And remember, that ability to go from NAD+ to NADH is reversible. It is not consumed. It is present all the time, and it can be used in a multitude of reactions. So stage 2 is where the energy is generated. We've expended energy in stage 1. Stage 2 is where the energy takes place. And those 3 carbon fragments ultimately give rise to pyruvate. This is one of the key molecules that you need to remember. Okay, pyruvate is there. It's the output. What happens to the pyruvate? Now, my aim is giving us good detail, and I've given you more of a global picture, more of a global overview of some of those steps. Okay, now, this next question is really talking about the fate of pyruvate, and what we can see here is alternate pathways. And we say alternate pathways because reactions can occur either in the presence or in the absence of oxygen, and also in different organisms based on the presence or absence of that oxygen. So if we think about that, this is really going to answer this question. So what do we think? What are the different pathways for pyruvate, thinking about presence or absence of oxygen, and also the, if you think in a human perspective, you might be thinking, well, where is there not going to be an oxygen molecule available? Think about exercise. Think about where there's strenuous exercise and the level of oxygen perhaps starts to decline. So anaerobic is ethanol lactate, aerobic is acetyl-CoA. Perfect. So can be converted to ethanol lactate, which is both, and both of these are fermentation events, and it can be metabolized fully to give rise to CO2 and water. So this is what we're going to spend a lot of our time focusing on later. The ethanol lactate occurs in the absence of oxygen. So this is a different divergent pathway. So if we don't have oxygen, we still want to generate products that could be of use. And so this is what occurs within the host. And we, of course, have used or exploited this reaction for the ethanol to generate a product that is favorable. In the presence of oxygen, full metabolism. It goes into the metabolic pathway, and CO2 and water are the end products. This is the main reason we need oxygen. The main reason we need oxygen is it is a key component of taking sugar, metabolizing it, and generating ATP, right, through the citric acid pathway, going into oxidative phosphorylation. Oxidative phosphorylation is where oxygen is involved. Oxygen is part of the reaction, and the phosphorylation is what leads to the energy or the generation of ATP in the mitochondria. So it's that whole process that is critical. Okay, so enzymes. Why are enzymes catalyzing irreversible reactions, potential sites of control? And I've briefly touched on this before. What do you think? Mainly, activity of enzymes can be controlled through allosteric effectors to increase or decrease activity. Great. So remember, you shouldn't think of an enzyme like a switch where it's on and off. What you should be thinking of is that it's like a dimmer switch where you can turn it on, and then if you need more activity, you've got ability for molecules or substrates or products to come along and stimulate the activity, so we're turning the dial up. And then as those products increase, they feed back, and we're turning the dial back down. We're then turning it off and silencing it off. So it gives us that control. So irreversible stops the flow of molecules going in the opposite direction, and therefore that becomes another point of control. It's like a de facto rate limiting step. It's another point within the cascade. So if you've got one that starts at the beginning and you keep going, that's a nice way of affecting the entire cascade. But if you've got another point which is part way through and it may diverge at that point, it gives you another point of control, another way of regulating the activity. So glycolysis, we've got phosphate fructokinase, PFK, hexokinase, and pyruvate kinase. These are all irreversible reactions. These are three molecules that you must know, because these are so critical for this process. And as we just heard, they can, through the allosteric effectors, can increase or decrease activity. And I gave you lots of different diagrams, lots of different examples, and we'll talk about PFK below, where the product can come back and either turn it on or turn it off. So glucose can help stimulate the activity. So if you've got a lot of glucose and you need energy, you want to stimulate the activity of these enzymes. Similarly, if you've got lots of ATP, that's suggesting that there is already a lot of energy reserved within the cell. There's not much point in going back to the cell and saying, well, let's take all this glucose and make further ATP. You'd want to divert it going elsewhere and sort of stop that from occurring. So this is where you might want to store the glucose. So if I go back to my example of the aluminium, if you've got 1 million aluminium cans in the factory and you've only got an order for 100,000, you've got 900,000 aluminium cans sitting there. You're not going to go out to the factory and say, let's mine some more ore to make lots of aluminium. You would wait until you get to a point where actually we've got a big order, we're going to have to make some more. So you need to be agile, you need to be able to respond accordingly. And so what is in the store is like your product. Your activity in the factory is the enzyme, and that product directly controls the activity of the enzyme. And it's the same sort of idea that we've got here. So this is an example of 3 enzymes that you do need to know. You do need to know what they're doing. We've spoken about hexokinase above. Pyruvate kinase, well, you can tell what it's going to do. It's going to lead to phosphorylation of pyruvate. And phosphate fructokinase, again, we've spoken about earlier as a control point, but we're also going to talk about it again here. Now, we focus on PFK because it is one of those irreversible reactions, but also because it plays a key role in the liver and muscle. And these are going to be the 2 points that we're always going to compare. Liver activity, so the role of glucose within the liver. So remember, the liver is important in controlling the level of glucose within the blood. And muscle is where you have to use glucose as an energy source. You're going to run, you're going to do something. You need that as a source of energy. And remember, you've got glycogen as that short-term store, and then you have to find alternate ways to feed energy into the muscle. So how does the control differ between the two? So, as I've highlighted again, PFK is the most important control point in the mammalian glycolytic pathway. This is why I'm highlighting it to you, to say you need to know what PFK does. This is one of those key points. Remember, if this is the key point, why is it important? Because it controls the pathway. It controls what's going on. This also then highlights why we're not going to spend as much focus on some of the other enzymes that are perhaps following some of those subsequent steps, because they are all dictated by what PFK is doing. That is the more important aspect. As I mentioned before, if you've got high levels of ATP, it will inhibit PFK. And there's a lovely diagram, which I'll show you later in your notes, that shows the shape in the absence of ATP, and in the presence of ATP, the whole curve is shifted significantly to the right. You need to have a lot more before you start to see any activity. So energy demand is low, so the enzyme has to be deactivated. Why is it low? Well, energy is ATP. If you've got lots of ATP, there's no need to make it, right? You want to turn off the enzyme. And the other example that we had was about the acidity and the pH. And this is in those anaerobic conditions. So you've run, you're breathing heavily, you're starting or you're exerting at a high rate. The level of oxygen that's coming in is, I don't want to say insufficient, but it's not enough to get to your muscle to be exchanged effectively. And so you start to still have that energy expenditure, but rather than operating in aerobic conditions, you're now operating in anaerobic conditions, and lactic acid starts to build up because you still have to have those molecules being generated. This will also inhibit PFK because as we start to get acidification, you don't want to have muscle damage. You don't want your muscle starting to become more and more acidic. That's going to lead to a whole raft of issues within the tissue. That's the basic idea. So now let's go back to the question and talk about liver. So I put that there just to give you the baseline to understand the basic points. So with the liver, those 2 factors, ATP changes and pH, are less important for controlling PFK because its role is more about glucose within the blood, right? You're not going to have, the liver is not going to be there energetically contracting. It's not a muscle, right? So that aspect is not going to occur. What you're going to find is that PFK is going to be inhibited by citrate, which is an intermediate of the citric acid cycle. This is very similar to the ATP, but all we're saying here is that these molecules go into the citric acid cycle. The output of the citric acid cycle are intermediates and other cofactors that can lead to ATP generation. So really, it's just an alternate way of sensing high levels of energy. And in this case, high levels of intermediates that feed into the pathway that give rise to energy. So really, it's very similar, but it's just that they're coming from a different location. So citrate feeds back and inhibits. Notice it's an early intermediate. So again, it's one of those first steps in the TCA cycle. Now, it's obviously a cycle, it's circular, so there's multiple points of entry, but things feed in at one point before they come through. This is where citrate feeds in, so it's one of those early steps. So high levels of citrate indicate high levels of biosynthetic precursors, so additional molecules are not required. It is the same logic. If you've got a high level of this, you've got all the molecules that feed and generate this must also be present at a high level. There's no point making them. We don't need to then turn on the PFK so that we can stimulate and give us additional molecules. It would have to be the opposite. We want to turn it off and inhibit that. So liver responds to the changes in blood glucose through that fructose 2,6-bisphosphate, which can stimulate the activity. And in the liver, fructose 6-phosphate, so remember the precursor, as these levels rise, when glucose levels rise, it activates PFK, which stimulates glycolysis, because remember, it's part of that process that we're going through, fructose 6-phosphate to be converted to fructose 2,6-bisphosphate. So again, it's all about the key molecules. What is the function of the organ? When you look at these sorts of questions, just think about, what's the liver doing? What's the muscle doing? Liver is involved in glucose levels, so it's going to be regulating the level of glucose that is released into the bloodstream. That's it. It's not going to do the same thing. Its requirement is going to be different. In muscle is going to be different because it has to expend energy, it has to contract, and it uses this as an energy source. So therefore, what changes is going to be different, and that's where the ATP, it's where that acidification comes into play. Okay, so that stops the finishes there, but let me quickly go back to you as a group before I go back to the lecture, and I want to highlight those key points to make sure it's really clear and addresses that comment I made right at the beginning about the importance of the chemistry and really where I highlight the key aspects. So again, either in the chat or as an emoji or a thumbs up or something, let me know, because these ones are going to start getting more and more complicated. And essentially all that happens is that they can get converted. Diagrams are including the chemistry, because if you're good at chemistry, you like to see where things are occurring. That's why I've put them in to show where those structures are, what the groups are. But really, to me, the key point is, what's the first step? First step is hexokinase. What is it doing? It's taking a phosphate from ATP, converting it to glucose 6-phosphate. The net result is that glucose can no longer exit. It is fixed into this pathway, and it now becomes the substrate for this first stage. We saw the isomerization, so really just converting glucose to fructose, just moving groups around. Remember, it's an isomer. Phosphate fructokinase, we add it again, and then we convert these down to intermediates. So this is the aldolase. Remember, an aldolase, again, is like an isomer. It's converting the dihydroxyacetone into, well, this is the isomerase here, but it's generating these two products, and then there's an isomer that converts it to glyceraldehyde 3-phosphate. So really, glucose, glucose 6-phosphate, and this one here are the key ones to be involved and understand. Remember that we've got two molecules here, but this one here, the DHAP, is converted to two molecules of the glyceraldehyde 3-phosphate. So that's why when we come to the second stage, we've got a ×2 that is being shown, because two molecules are entering, not just the one. So this is an important point to highlight. We're getting two products being generated. Now remember that when we were going through, I think it was Mayuni had the great detail about the individual enzymes, and I really glossed over those, because it's really probably this last point, pyruvate kinase, which generates your pyruvate, and remember that pyruvate is then the key molecule that's going to move forward. These other steps are just sequential processes. And again, what are we seeing? Well, we've got NAD going to NADH, that's important. We've got more use of, we've got the regeneration of ATP, so we used it up here, but we've regenerated, so they help to balance each other out. And again, down here, we've generated another molecule of ATP. So again, above in that text, we had glucose and ATP generation, we had the quantification of that. So that was the diagram. Then in the lecture, we go through into more detail saying what are those exact steps. And I've highlighted to you what I think are really the key points. Phosphorylation by hexokinase, generating glucose 6-phosphate. It can't exit, and this is the reaction. And notice the forward reaction is by far the more preferred option, because it's a hydrolysis of ATP, so you want it to go in that direction. We've got the isomerization of glucose to fructose. We can see here, we've got 1, 2, 3, 4 carbon atoms. This group here, and that group is, to you, what I think other steps are just by far the more preferred option because it's a hydrolysis of ATP, so you wanted to go in that direction. We've got the isomerization of glucose to fructose. We can see here, we've got 1, 2, 3, 4 carbon atoms. This group here, and that group is just shown in a different position, so it's that changing of the location. We've got another phosphorylation, and this is where PFK comes in. So this is that process of phosphofructokinase. Remember, this is that important molecule, and again, notice the forward reaction because it again is another ATP-dependent process. We've then got the cleavage of those groups into the 3 carbon fragments, which are glyceraldehyde 3-phosphate, dihydroxyacetone phosphate. Don't worry about the names. If you have to remember one, remember this one, the gap, because remember the DHAP is converted back into the glyceraldehyde 3-phosphate. Again, it's another isomerization reaction. And we can see that here. Chopped, and then these two get converted. This gets converted back to this one. So all products can be used by glycolysis, so that means that we generate an additional molecule. So net result is that 1 molecule of glucose, remember this is just for 1 molecule of glucose, converted to 2 molecules of glyceraldehyde 3-phosphate, 2 molecules of ATP have been consumed. So we have energy input into this system at this stage. Stage 2 is where we get the energy back out as well as other molecules being generated. So we see the NAD+, we see phosphate again coming in, so this is just showing us again ATP. And look at the enzyme that is involved. Even if you don't worry about the first part, look at the second part, dehydrogenase. D is telling you that it is depleting, it is removing. What is it removing? Hydrogen. And we can see that we've taken hydrogen away and we've added it to NAD+, as well as a proton. Remember we were talking before that when you start to generate these reactions, you can get acidification, and that's what this proton is doing. So this is again another good example of a redox reaction. It's the removal of a proton, and it is being transferred to NAD+, to NADH. So don't lose sight of the fact of these molecules that are involved. So it's reduced to NADH. Now, this molecule, 1,3-bisphosphoglycerate, is important because it's energy rich. It's got phosphate group, it's got double bonds. It will be used to generate the ATP. And so this is what gives rise to those 2 molecules. And then all of these other steps are really that processing. And we see the ATP, and it's just showing you what's the first step, what's the second step. Have I really focused on these individual steps and saying that these individual steps are critical? No. Does that mean that I expect you to know these individual steps? No. I want you to focus on the key enzymes, the key points of control, and really, pyruvate kinase, which ultimately gives rise to pyruvate, that is probably the more critical one because that is the point where we've got the pyruvate, which then feeds into the next pathway. These are all important. Are they important for us? Not so. Okay, the focus is more big picture. Hopefully that makes sense. So we can see the net reaction. So this is taking stage 1 and stage 2. Glucose, we've got 2 phosphate molecules shown, which means that 2 ATP molecules have been used. We've got 2 ADPs and NAD+, and we generate 2 pyruvate molecules, is important. We've generated 2 molecules of ATP. So yes, we've got some ATP, but remember, this is not the bulk of the ATP. This process is not designed to generate a lot of ATP. It's designed to generate precursors. Those precursors will then enter other pathways where we get the bulk of the ATP. NADH, acidification, and some water. Things are just balancing. Okay, so for this to occur, and we see that here, NAD+ must be regenerated as it's present in limited amounts. Again, I've highlighted this. This is an important point. Remember, NADH and NAD+ are interconvertible. You can lose the proton, you go back, you gain a proton, you bring it forward. It's recycled. So it's regenerated because there's not a lot of it. And because there's not a lot of it, it becomes, again, another point of those critical control points. So again, highlighting a lot of similarity in what we've seen. And then the pyruvate, where does it go? Well, if we don't have oxygen, or if we're in a plant, for example, or a yeast, goes to acetaldehyde, comes into ethanol. Again, you can see other molecules being generated. And we know that ethanol, for example, comes from milk, from CO2. You can see that there. In mammals, it can go straight to lactate, building up lactic acid. And in the presence of oxygen, again, we generate CO2, which we expire, but we also then make acetyl-CoA. And acetyl-CoA, further oxidation. And this is, again, the next precursor that goes into the citric acid cycle. And again, highlighting here, notice the regeneration of NAD+. So this is important in this pathway. It's being cycled and recycled throughout. So we see that here. NADH, stripping off the proton, going back to NAD+. Remember, it can then go back to the beginning, take the proton, and then be regenerated. So, full energy release. Pyruvate goes into the mitochondria, converted to acetyl-CoA, enters the citric acid cycle, and this is where we generate the large amount of ATP. So notice the change in focus again. Other sugars, as I mentioned, we start off with glucose. You've got galactose, fructose coming in. They come in at different points, so they can be converted. So galactose can be converted to glucose 6-phosphate, fructose to fructose 6-phosphate. It's the same thing. And then you get these intermediates. So again, it doesn't matter what the sugar is, it can be fed in. We focus on glucose because it's the starting point, but it's by no means the only sugar that comes in. Okay, so the control of the pathway we've spoken about. This is the diagram that I was referring to. I just need to zoom out a little. we've got velocity and fructose levels. You can see that in low levels of ATP, we want this to proceed. You can see that it's got a normal shape going up. When you've got a lot of ATP, notice the depression, notice the gap. There's quite a difference, particularly at very low substrate levels. It's only when you get at very high levels does it start to occur. You've got the key enzymes, so we've spoken about PFK, we've spoken about hexokinase here, and pyruvate kinase. There's three that I was saying to you, you must know, and the importance of those. And again, we talk about muscle, so at rest, where glycolysis is inhibited. Why are we inhibiting glycolysis at rest? Because we don't need a lot of energy. Notice the gap, there's quite a difference, particularly at very low substrate levels. By no means the only sugar that comes in. Okay, so the control of the pathway. We've spoken about, this is the diagram that I was referring to, I just need to zoom out a little. We've got velocity and fructose levels. You can see that in low levels of ATP, we want this to proceed. So you can see that it's got a normal shape going up. When you've got a lot of ATP, notice the depression, notice the gap. There's quite a difference, particularly at very low substrate levels. It's only when you get at very high levels does it start to occur. You've got the key enzymes, so we've spoken about PFK, we've spoken about hexokinase here, and pyruvate kinase. There's three that I was saying to you, you must know, and the important. And again, we talk about muscle, so at rest, where glycolysis is inhibited. Why are we inhibiting glycolysis at rest? Because we don't need a lot of energy. So why make a lot of energy if the muscle is not doing anything? If you're asleep, do you have a high energy demand on your muscles? No. But if you went for the run first thing in the morning, then you're going to need that energy, and then we go across here, we want to stimulate glycolysis, and we go through this entire pathway, so pyruvate, etc. Right? So we want to feed in, and we want to turn things off. How do we turn it off? Well, ATP, see here, ATP is shown as larger because we've got energy reserves, comes back and inhibits, and similarly glucose-6-phosphate inhibits hexokinase. On the other hand, when energy is low because we've started to run, AMP levels are increased, this stimulates PFK. Makes sense, it's the opposite, we inhibited here, so if the other molecule is elevated, it should stimulate, and that in turn then feeds forward to again generate additional molecules downstream. So again, hopefully that all makes common sense. We spoke about the control in the different pathways and the liver versus the muscle. We spoke about feeding forward and feeding backwards. And again, these are probably a little bit more complicated diagrams, but again, showing you the same thing, that if you don't have, diagram showing fructokinase activated by low levels of fructose 2,6-bisphosphate. You can see small changes, 0.1 micromolar, but look at the shift. And similarly here, with ATP initially stimulating, but again, you don't need massive amounts. So these levels are really important. 0.1 micromolar. Think about that, it's really quite low. Charlotte, so can ATP and AMP both inhibit and stimulate? Yeah, so let's go back to here. In this example, depending on what is occurring, it's pyruvate kinase in those different areas. Don't compared to the muscle. You're going to be bad for about what was. Okay, let me just stop there and I'll stop sharing. Oh, thanks, Ira, Charlotte.